Hey guys and girls, hi everyone. So you are here with our next speaker. I'm very glad and honored to introduce Dr. Jonathan Karr. He is well known because of the wholesale model. Previously, with Alexei Kolondik, we talked about the concept of virtual human. It was quite interesting, but you have to keep in mind that we need somehow also to make a kind of checklist. If you know where I'm going, as Alexei Kolondik nicely brought to us as a discussion, but where you are now, I mean, Jonathan Karr, he did a very nice model. It's called the wholesale model. I was very glad to attend to his talk like twice. The first time I was very surprised how friendly he is, like move and interact with people. I'm very curious to see how he's going to manage this new era of online because it's a little bit harder for him to interact with people online. So let's see, let's see how he adapts to this new reality, this new realm. So I will not talk too much. I'll just make some quick announcement here. First of all, now you have the possibility of donations if you want to help the channel somehow. This is will be the last live from this set of live I'm give. I'll give a short break. I mean, I don't know. I'm not sure yet how long will be the break, but I'm planning to make the next lives like a theme, like we choose a topic. So you are welcome to let me know which topic. I didn't decide yet which topic will be, so I'm thinking to organize people around the topic, choose a topic, and ask people to talk about that. This one was a little bit the first sequence of live of the channel. As I said to you, I start to make live on this channel. And as always, you have the option to talk on the chat. You can send your question. Your question will appear here as a kind of a contribution to our discussion. And feel free to ask anything that we are going to do our best to answer your question. So please, Jonathan, you have the word.
All right, well, thanks so much for the opportunity to share the work that we've been doing to accelerate the development of wholesale models. So I'll go ahead and share my screen. Okay, so I was asked to start by speaking about my own personal journey to wholesale modeling. And so I am a principal investigator, faculty member at the Icahn School of Medicine at Mount Sinai in New York. As was introduced, the focus of my research is on developing more comprehensive models of individual cells, and the technology that I think many others believe will likely be required to realize such models. In addition, I mean, I personally feel like technology will need to play a major role in realizing wholesale models. At the same time, I think that a large number of people will need to be involved in the development of wholesale models, and for that reason, I have been and continue to be involved in trying to foster the interdisciplinary community of scientists and engineers which will be needed to realize these models. In the interest of kind of explaining who I am, I'm also a co-director of a PhD program in genomics and data science at the Icahn School of Medicine at Mount Sinai. So my attention is primarily focused on two categories of projects right now. Presently, we have one major modeling project, which is to develop an integrated model of one particular human embryonic stem cell line. In addition, we have a couple of smaller projects to try to expand a metabolic model of E. coli and develop a model of the metabolism of individual mycoplasma pneumonia cells. And then, as I'll speak more later in the talk, we are developing a variety of software tools and computational methods behind those tools to enable more comprehensive models of individual cells. So how do I spend my time? Most of my time is spent developing the models that I illustrated on the previous slide and the tools for modeling that I mentioned on the previous slide. So that involves things like gathering and assembling, integrating, normalizing data, designing models, combining models of different cellular processes, designing simulations of these models, calibrating the models, trying to run simulations, compare them to data to prove that models make accurate predictions, creating data visualizations to help us assess this, and then designing data structures, algorithms, softwares to implement and manage the data, models, simulations, visualizations, and so on. As a principal investigator, I also spend a portion of my time managing the other individuals who I collaborate with, disseminating our work and applying for and managing funding to support this research. So how did I get interested in wholesome modeling? So from a fairly early age, dating back to high school, I was interested in medicine and physics and math and computation. And at the same time, I also I grew up not far from where much of the human genome was sequenced, and so I think from a fairly early age, I was influenced a lot by developments in genetics and genomics, and then more recently, the availability of big data in biology, particularly in genomics. And so as a high school student and then early in college, I spent a lot of time trying to explore this intersection, trying to explore the intersection of my interests. And one of the first areas that I spent a lot of time investigating, and in fact, as a college student, I actually majored both in physics and neuroscience. I spent a lot of time exploring computational neuroscience as an undergraduate student. And at the time, I had been aware of systems biology, but it was a newer field, and so I didn't have quite as much opportunity as an early undergraduate student to explore systems biology. So this was in the early 2000s. At that time, systems biology was relatively new. For example, at that time, I was an undergraduate student at MIT. At that time, MIT was just starting what's now called the Computational Systems Biology Initiative, which has come to be mostly a graduate program in computational systems biology. At that time, MIT was really just starting the SISB initiative, and there weren't any classes really yet. There still is no undergraduate program associated with that. Harvard, which is nearby, was also just starting to get their systems biology department off the ground. And so finally, at the end of college, I had an opportunity to take a course in systems biology from Alex Van Unenarden. And I really enjoyed that course, and that really convinced me that I should focus my attention rather than on computational neuroscience, but on systems biology instead. And there were several reasons why I really liked systems biology. It's a very similar field, I think, to computational neuroscience in a lot of ways. But I felt that systems biology was a field where the scale of the problems is such that it's possible to have to build mechanistically motivated models that tie back to physical chemistry, that tied back to knowledge of dynamical systems. And at the same time, there's a large amount of experimental data, principally from genetics and genomics, that we can use to try to build mechanistic models. So it was, I think, a scale at which it would be possible to build data-driven, but yeah, mechanistically motivated, if not mechanistically accurate models. It was also an area which at the time, and I think is still true today, had a lot of opportunity, a lot of open challenges, and therefore, for a young scientist, a lot of opportunity to make an impact on science. So by the time I started graduate school, I knew that I was interested in building models of biochemical systems, and I had then the fortune of working with Marcus Covert, who then challenged me to try to the extent possible build as comprehensive model as I possibly could of a single organism. And then over several years, Marcus and others and I assembled what later became one of the first whole cell models of Mycoplasma genitalium, which is a small gram-positive bacteria. And after graduate school, I became a faculty member at the Icahn School of Medicine at Mount Sinai, where I've been, over the last several years, trying to bridge from kind of very early stage models of entire bacteria, try and get us from kind of coarse models to much more accurate, much more predictive models, hopefully encompassing eventually even human cells. So I thought I would just reflect for a couple of minutes about what I felt is key to working in this field. So because whole cell modeling aspires to, within a single model, model a broad range of biochemistry, I think it's essential to have a pretty broad understanding of molecular cell biology, biochemistry, physical chemistry, and dynamical systems. And then I feel that whole cell modeling and maybe systems biology in general is an area of science which is still relatively immature, in the sense that there's not a lot of established methodology, established software tools, established resources that a modeler can easily just follow. And so I think to be productive in this area of science today, I think that requires having strong skills in computer science, specifically in software engineering. And if I look at the people that I've worked with, which I think have been the most productive, I think they've generally had very strong skills, not only in writing code, but also in testing code, in continuously integrating code, and using source code management systems like Git to be able to work effectively with collaborators and not only write code, but quickly and effectively write code that really works as you intend and can be combined with yet other codes for other problems. So I think beyond just those skills, I think in my opinion, working in this area, I think requires a certain personality or certain interest in particular topics. So I think from my perspective, one of the key defining things about whole cell modeling is that it's a field with a lot of big goals and a lot of big challenges. And so as a result, I think working in this field requires one to be able to take a big long-term goal and then to be able to translate that into small steps which are actionable in the near term. And then be comfortable with the fact that the solutions that you're going to try to build to relatively near-term problems over the short term, that these may or might not actually. I think you have to be comfortable with the fact that wholesome modeling is an emerging problem which doesn't have an established set of tools or established set of methods or protocols that one can just follow. I think you have to be comfortable working in a field that has a lot of long-term uncertainty. And because there aren't a lot of established methods, instead I think it's important to be able to identify relevant methods, relevant tools in other established fields, and to be able to bring them into wholesome modeling, learn from them, adapt them, and combine them as necessary to advance the field of wholesome modeling. And again, very, very similar to that, I think, because there are many challenges in wholesome modeling, in order to advance the field as a whole, I think one has to, it's important to have the ability to and willingness to work on a broad range of tasks. So, for example, if you want to build a big model of a new cell type, you'd have to integrate the data, find the data required to do this, integrate it. You have to design a lot of submodels. You'd probably have to write a lot of software. And so I think it's difficult to make progress in wholesome modeling and want to be siloed. I think it's important to be able to want to work broad across a range of tasks and push all of them forward to realize bigger long-term goals. Okay, so at this point, unless there's any questions, I want to transition to talking about some exciting work that we've been doing to try to accelerate the construction of wholesome models. So from our perspective, we think that the future of medicine and synthetic biology will likely require much more comprehensive models of the relationship between genotype and phenotype. One of the main reasons that we think that more comprehensive models will be necessary is that from sequencing individuals over the past couple of decades, we know that every individual has a unique set of genetic variation, which is scattered throughout their entire genome. We also know from a variety of evidence that this genetic variation affects how people affects physiology, affects disease physiology, and it affects how individuals respond to drugs. So, for example, we know that two individuals which appear to have the same disease phenotype, they may not respond the same way to the same drugs because their underlying genotype differs. And so this has, of course, led to the realization that we need to be able to select for each patient the best course of therapy or predict their most likely disease trajectory based on their unique genetics. And so this is where we think that more comprehensive models come in order to be able to deliver precise care to individuals, or at least be able to make precise predictions about the disease trajectory of individuals. It seems necessary for us to have models of entire genomes resolved down to the level of individual nucleotides. And I think this need for models which are both broad in the sense that they cover the whole genome and deep in the sense that they represent, they allow us to reason about individual nucleotides is a sort of an inescapable fact of medicine. And very similarly, we think that much more comprehensive models of entire genomes are going to be essential to the realization of synthetic biology. Very similar to other engineering fields, we think that the bioengineering will likely require computer-aided design tools, and that this in turn will require comprehensive models of entire genomes. So more specifically, we believe we have to build models of entire cells we have to build models of their whole genome resolved down to individual nucleotides. These models have to capture the entire behavior of a cell, including its behavior over its entire life cycle. And for a variety of reasons, we feel that it will likely be important to build mechanistic models. For one, we think that precision medicine will likely require mechanistic explanation. If we're going to use models to recommend therapy to individuals, we think that it will be important to be able to provide a mechanistic explanation of why that therapy should be delivered to an individual person. And so for a variety of reasons, then what we're talking about is building dynamical models, which will be able to capture the dynamical behavior of cells over their entire life cycle, and models which are also stochastic and therefore able to capture the variation among the phenotype of individual cells. So a few years ago, we began to ask ourselves a simple question, which is, to what degree is it possible today to build more comprehensive to build a comprehensive model of an individual cell? And that led to constructing what's now considered one of the first whole cell models of a cell, where we focused on the small gram-positive bacteria Mycoplasma genitalium. And what we were able to show is that even with the data and the methodology that we do have today, that it is possible to build relatively comprehensive models, albeit of very small organisms with small genomes. Now, we created this model by integrating a broad range of data about Mycoplasma genitalium, gathering this data from a variety of different sources, organizing it, normalizing it, and integrating it together into a relational database. We then built software to turn that data into a set of submodels that represented different biochemical subsystems in Mycoplasma genitalium. We then wrote our own software we decided to take a modular approach to constructing the model, in which we design the model by assembling a set of submodels that represent different cellular subsystems. And then furthermore, we realized that we could represent these subsystems using different mathematics, which were appropriate to the physical scales relevant to each cellular subprocess. So then we developed codes to be able to co-simulate these submodels, which were described using different mathematics. We then further developed tools to be able to calibrate these models, to organize large sets of simulations and visualize them and analyze them to extract insights in them, and to determine whether we felt that they made accurate predictions about the physiology of Mycoplasma genitalium cells. And so this graph illustrates the comparison between the predicted growth rate, between the observed growth rate of Mycoplasma genitalium in blue for disruptions of all of the genes that are listed at the bottom of the graph. So each kind of column of this graph represents the observed and predicted phenotype of Mycoplasma genitalium upon the deletion of one individual gene. And so the blue represents the experimental measurements, and the red represents the model predictions. And so what we find is that in most cases, the model is able to quite closely recapitulate the observed, the experimentally observed phenotype of Mycoplasma genitalium. But at the same time, we see that as comprehensive of model as this is, there's a number of genes, and those are the ones that are highlighted at the top, which when we delete them, the model is really not able to well capture the growth phenotype of this organism. And so what we took away from this is that while we did demonstrate the ability to feasibly construct a model of an entire cell that represents many biochemical systems in that cell, we still have a lot of room for further progress to build more accurate and also more comprehensive models. And so over the last several years, we've been thinking about how do we get from this primitive ability to build relatively coarse or comprehensive models that make still relatively coarse predictions about the phenotype of cells. How do we scale this up to much more predictive models, which encompass much more biochemical activity? And furthermore, how do we do that for yet more comprehensive cells with vastly larger genomes like human cells? In order to do that, I think we have to work through a number of challenges. We have to be able to explain simultaneously within a single model a diverse range of biochemistry. We have to be able to, within a single model, describe or capture the many different forms of proteins which exist out of a combinatorial set of processes that add enormous complexity to biochemical networks. We have to do this across a number of different scales. And on a more practical level, we have to do this despite the fact that we have no single way of experimentally characterizing a single cell. And instead, what we have to do is assemble or attempt to assemble a comprehensive understanding of a cell by combining together multiple experimental modalities. And another practical challenge is we have to do this in spite of the fact that most of the data that we have to possibly construct models of cells is incomplete. So a typical mass spectrometry metabolomics experiment, for example, although there's thousands of metabolites inside a cell, we can maybe at most observe 100 or a couple hundred metabolites in a single experiment. And even then, there's only a few examples of data sets that are even that large. And so I think one way to look at it is that every aspect of this workflow that we pioneered to build a wholesome model of Mycoplasma genitalium, there are challenges to operating this workflow at a much greater scale, and we have to try to develop technological solutions to each of these major bottlenecks to progress. So for the next few minutes, I'll tell you a little bit about several methods and software tools that we've been building in order to accelerate this workflow and in turn more comprehensive models of cells. So a couple of years ago, we ran a survey of the biochemical modeling community and related modeling communities, and what we found, and one of the questions that we asked was, what do you feel is the most critical bottleneck to progress in biological modeling? And the most frequently cited reason was lack of access to the data that modelers felt was essential to building models. So let's take one example. Let's say, for example, we're interested in building a dynamical model of metabolism. We would very likely need data about concentrations of metabolites, data about abundances of enzymes, data about rate constants of chemical reactions. And if we wanted to model an entire cell, of course, the situation is much worse. We need data about, for example, the sequence of genome, locations of genes, locations of promoters, terminators, rates of transcription, rates of translation. And the key thing to understand is that this data isn't just available in one particular place. That this data is scattered in many different databases and many different articles. It's described with different units. It's described with different identifiers. It's gathered with different methods, with different machines, different protocols, different organisms, different environmental conditions. And so while there is a lot of data, it's extremely heterogeneous. And that heterogeneity makes it really difficult, ends up occupying a lot of modelers' time and preventing a lot of progress. So we developed a toolkit called Datanator to try to accelerate modelers' paths through this data, to help modelers find the data that they need, or that data that would be helpful to modeling projects more quickly. Datanator is available at datanator.info. The central underlying feature of Datanator is an integrated database of several of the most essential types of data required for this kind of biochemical modeling. Datanator includes, for example, data about concentrations of metabolites, modifications, half-lives, and localizations of RNA, abundances, modifications, and localizations of proteins, rate constants of reactions, and more. And each of these types of data is integrated from different sources. And furthermore, most of these kinds of data are actually integrated from multiple sources. So for the first time, Datanator provides a single place where this broad range of data can be accessed. And not only have we put all this data into a single place, but we've also normalized all the data, so converted it into a consistent set of identifiers and units to make this data easier to navigate. Now, as for any modeling project, modelers probably are typically interested in data about a specific metabolite or gene or reaction in a particular organism in a particular environmental condition. And as I mentioned before, these data sets that we're working with, they're typically incomplete. So while there may be data about a lot of metabolites, there isn't necessarily data about the specific metabolite that someone is interested in the particular organism and environmental condition that they're interested in. And so what is often done informally is that modelers will take advantage of related data about similar metabolites, similar genes, similar reactions, but this is done ad hoc. So in order to allow modelers to find data that's relevant to their problems quickly and at scale, we've developed some tools to help modelers navigate this sea of data that we've gathered into Datanator. So the first part of this search workflow is a full text search, substantially similar to other databases. Modeler can search for metabolite, reaction, or gene by a description, textual description of that entity that they're interested in, and they can specifically, if they want, indicate that they would like data in a particular gene in this particular taxon. Modelers are then presented with a list of metabolites, genes and proteins, genes and reactions, which match their search query. Then modelers can click on any metabolite, gene, or reaction, at which point they're presented with clouds of data, which we've assembled, which encompass not only the selected metabolite or selected gene or selected reaction, but and data in the particular organism that the user has selected. These clouds of data also encompass data about related metabolites or genes or reactions in related organisms. So essentially what we've done is what we try to do is pull out a cloud of data centered on particular point in this multidimensional space of molecules and reactions and organisms and environmental conditions, pull out a cloud centered on a point that the user is interested in. And then we provide users tools to be able to sort of filter down these clouds to just the very most relevant measurements to the user's study that they would like to execute. And then Datator provides investigators the ability to export this data in a structured format that's conducive to further modeling. So we feel that for the first time, Datator really makes it substantially easier to gather the data that's necessary for wholesale modeling, reducing one of the big time bottlenecks to constructing wholesale models, and opening up the ability for modelers to better leverage data from related organisms and related molecules when inevitably there's no direct measurement of the data that a modeler is looking for. Now, one of the long-term challenges to continuing to integrate data that's required for wholesale modeling is that much of the data that we need for wholesale modeling is published ad hoc, often as supplementary spreadsheets to journal articles. And in contrast, as modelers, what we usually want to work with is some kind of structured, rigorous, well-tested representation of the same information, rather than spreadsheets which are structured ad hoc and really oriented more for human readability. So in order to work toward making it easier for investigators to contribute data to Datator, we've developed a toolkit called object tables, which allows us to easily connect these two worlds of human readable spreadsheets and structured data objects. So object tables is a framework which allows us to define schemas for human readable spreadsheets and then use those schemas to automatically parse human readable spreadsheets into structured data objects that we can easily do further computation on, or easily translate into a schema that can be deposited into a database like Datator. So we feel that that object tables will be key to allowing us to continue to expand the content of Datator by ultimately enabling experimentalists to be able to contribute data directly to Datator. As I mentioned, we surveyed the biomodeling community a couple of years ago, asking the community, what do you feel are the most key bottlenecks to progress in biomodeling? And one of the next biggest bottlenecks was cited was the lack of clear descriptions of complex models. In the field of systems biology, there have been several model languages that have been developed which are capable of transparently capturing comparatively simple models, but there's really never been a commonly adopted format that allows for transparent descriptions of much more complex models, which might be composed of submodels, where each submodel may be represented using different mathematics. And so over the last couple of years, we have begun to develop a language that we call WC-Lang, which allows us for the first time to be able to transparently describe composite multi-algorithmic models, where a model is composed of submodels, each of which are intended to be represented and simulated with different mathematical representations. And one key aspect of WC-Lang, so as I mentioned, in building wholesome models, we need to be able to capture an enormous amount of biochemistry inside a single cell, and we need to be able to do this at a very fine-grained resolution all the way down to individual nucleotides. So let's say, for example, as part of a model, we want to model the transcription of individual nucleotides. One possible way to do that would be to enumerate every individual possible intermediate transcript in the course of transcription and every individual nucleotide elongation reaction. And so that would lead to this enormous number of reactions which no one would possibly be able to understand. And so instead, what I think we want to be able to do is recognize that transcription actually is a pattern. It's a template-driven reaction driven by DNA templates. And similarly, RNA degradation and translation and protein degradation, these are all template-driven reactions where there's a DNA template which encodes RNA, or RNA template which encodes DNA. And so, in fact, we could potentially express all of this biochemistry much more compactly using a small number of reaction rules. So a key part of the development of WC Lang has been the generalization of rule-based modeling to support sort of higher level rule patterns by generalizing rule-based modeling from languages like BioInformatician and Kappa. I think related to the development of WC Lang, we have also developed WC Sim, which is a tool for executing models which are described using WC Lang. Now, so as I've mentioned a few times, we feel that one of the most promising ways to build wholesome models, and what we have been pursuing, is to be able to combine or co-simulate multiple models of different aspects of cells using different simulation algorithms which are appropriate to the different scales relevant to each cellular subsystem. And so to make it easier to combine simulation algorithms together into co-simulations of multiple cellular subsystems, we have been working on several standards for simulation tools, which have allowed us to create a standardized set of simulation tools which can be more easily composed together into multi-algorithmic simulations. So these standards essentially build on Docker and SetML, and essentially what they allow us to do is, using identical syntax, be able to run any simulation tool and get out results in a consistent format. So presently, we have 18 simulation tools standardized as part of biosimulators, and each of these can be used in a standardized way, and in turn this makes it easy to, instead of just simply running individual simulation tools, it makes it easy to compose simulation tools together into multi-algorithmic simulations. So at biosimulators.org, we have a web interface for exploring the simulation tools which we've standardized. This provides detailed information about the capabilities of the simulation algorithms, the model formats, the model frameworks that are supported by each of the individual tools. So this sort of basically tells you the capabilities of each building block. The building blocks then behave in a standardized way, and then this combination of standardized behavior and detailed metadata provides a substrate for combining these simulation tools into multi-algorithmic simulations. On top of biosimulators, we also provide a web application called Run Biosimulations, which makes it easy to use these individual simulation tools to reuse published simulations. So the Run Biosimulation simply provides an easy-to-use web form where modelers can upload a simulation project, select a specific simulation tool that they would like to execute, a particular version of that tool, and then they can simply click a button and run simulations involving several different model formats and over 50 different simulation algorithms. Presently, we're working on another layer on top of Run Biosimulations, which will be called Biosimulations, which will provide a platform for sharing models and simulations across investigators. Okay, so finally, where is all this going? In our own lab, we're using this set of technology, this increased ability to find data, other tools that I haven't discussed for constructing models, this increased ability to transparently describe models and easily simulate them with WCSim. We're applying all of this to develop one specific model of a human embryonic stem cell. So far, we've started by assembling individual submodels of a human embryonic stem cell, building models that capture its metabolism, its gene expression, and its turnover. And we've now assembled those models into a single model and are working to validate the model and use it to drive novel biological insights. So to quickly summarize, we think that wholesome models are becoming more feasible, and in particular, we feel that the introduction of several technologies that address the principal bottlenecks to wholesome modeling will accelerate progress and ultimately enable much more comprehensive models, which hopefully over the longer term will enable physicians to make more precise medical decisions for individual patients and help engineers rationally design microorganisms for a broad range of possible applications. And finally, I just want to thank everyone in my lab and our collaborators of the last several years who've contributed to all of the work that I've shared today. So with that, thank you very much for your time and your attention, and be happy to take any questions.
Hey, Jonathan, thanks a lot for the amazing talk. I was like quite like amazed by all the most of the questions I had prepared. You already answered, so I'm just going because I thought what I could ask and so on, but somehow you already like answered. But just to keep the discussion going on. Maybe it's the Saturday effect that you said to me offline about Saturday. We don't have question here, but I have my own questions. In general, I want to speak so far the talks that I had to introduce the topic of innovative biomathematics, which is one of the core topic of the channel. But you made my life easier, so I'm going to start with that. Essentially, I would like to, because I have two channels. Another one is about the web application, web development. It has to do with my second postdoc. So my second postdoc, I was working with a web application for target therapy. So I developed a web application that could show all the target of your system biology like simulation. So maybe that I can somehow mixture the two public, the one from web application, the one from biomathematics. Can you talk a little bit more about this challenge of, because you did a very nice, exactly what I think, I'm like in my head. A web application is quite friendly. People can put the simulation. Can we talk a little bit more about this challenge of building this web application? Maybe give a little bit more thought about this difficulty and so on.
Sure. Well, let's see. Where should I start? I mean, I think the web application that we're building for modeling from a technical perspective, there's not much that's perhaps really unique about them from other web applications. So we're able to use the same web development technology that other people do for lots of other purposes. So Datonator, for example, is built using React, which is developed by Facebook, and that's one of maybe.
I use Angular.
So right, so we use Angular for different projects. So we use both, and those are two of the most popular frameworks for reactive development today.
But in the case of React, because people say, I mean, I guess it's not you. I guess you have a team that work for you, that is working with you, and so on. React was able to do, because React has some kind of. They say that some people claim that React is a little bit immature in the sense that you can build fast, but have difficulty to go on later. It has been easier for you to, because you build the application, but I thought you are trying to increase the application. You're trying to make the application each time better. Did you face some difficulty with React, or it's just a legend? It's not true that React is difficult to make it bigger later.
So we have used React and we have used Angular in two different projects. And I think it's fair to say that the project that we're using Angular, it is substantially more complicated, and Angular has facilitated the size of that. So I think if you compare the two, React, I think, is definitely easier to get started. It's definitely more flexible. But one key thing, I think, which is not so much the fact that like one is a less comprehensive framework than the other. Angular is type. Right? Angular uses TypeScript, and so it's typed.
Yeah.
That typing, I think, provides a huge advantage for the development of large applications. Just matching types, and then the errors that you get when types aren't matched, that avoids a lot of need for otherwise having to write a lot of unit tests.
Unit tests, you have to talk about that as well.
I mean, you still need unit tests, but it's really amazing. If you look at like React, like I think it's hard to get React to work well without a lot of unit tests. In contrast, our experience is that with Angular, I mean, it's hard to get by with zero, but you need a lot less because you can rely on the typing and the type checking much more.
Yes, but just because, let's say, when I start my postdoc, I was given the chance to build this application. So I did a very strong research about Angular, React, and so on, Vue, all the kind of possibility. One thing that people used to say that Angular would be very nice for you to go on later because you had talked about the unit tests and so on. But in the case of specifically of React, which kind of I mean, I know that the technical detail maybe is not very, maybe I don't know, don't have to talk about technical details, more detail, but which kind of library are you using for the unit test, integration test? I'm not from the React community, but maybe they want to hear as well because you are using, so maybe they want to know more about the unit test, integration test, and so
on. For React, we use Jest and Cypress. So we use Jest for unit tests, and we use Cypress for end-to-end tests.
Perfect.
And I don't know, I mean, there's other choices. I don't know that. I mean, Jest, I think we found is pretty easy to use. I mean, Cypress is powerful, but also kind of a pain to use. You know, I mean, if I was giving advice, if people are looking for advice, I would strongly recommend, like, if you can't, because running end-to-end test is much more touchy. Because when you run end-to-end tests, the testing framework is the way that Cypress works, the testing framework is much more isolated from your code, so you don't have the ability to actually inspect the your code the same way that you do. You're testing at like a arm's length, and so you have much more obtuse ability to actually test something. So for example, okay, you run a test with Cypress, the only thing that you can do is you inspect the result in HTML. You don't have the ability to directly test the output of a specific function. And so if you want to test anything, you have to like query your result in HTML document. You have to wait until the browser has rendered this. So testing it is a lot more complicated. If you can, where possible, I would encourage people to think about how do you refactor your code so that you can, instead of trying to test it through end-to-end testing, test it with unit testing with something like Jest.
Yeah,
So
just
factor in all the logic so that you don't need to do end-to-end testing.
just out of curiosity, so you're talking about testing. Are you using test-driven development or behavior-driven development? Are you using kind of methodology to develop based on testing, or you just make the test after the application is ready, or you make a methodology like TDD, BDD, DDD, all this kind of like test-driven development, behavior-driven?
Right. Yeah, we for the last project that I mentioned, which it's this really integrated set of projects that we call biosimulators and biosimulations. So there we're continuously releasing additional code as we go along, and so we're simultaneously adding more tests and releasing. I don't know that I don't think we really use like a strict test-driven development philosophy, but I think it's fair to say that we certainly do have tests integrated into the development. It's not like we build the application and then we write tests. We're constantly going back and forth between.
So it's TDD. So I use TDD, test-driven development.
Yeah, but it's not. We don't. At least in our own group, we don't.
Force.
I wouldn't say we follow a very strict test-driven development paradigm.
Okay, but some people I have seen recently several talks from web developers. So do you agree that test is very important? Because today have two kind of developers: people that don't like test, people that like test. Some of them like they shoot each other, like as always. In your case, do you think that the test, because in order to create tests, it's extra work. You have to do extra work to make the test, and so on. Your code sometimes 3 times bigger than the conventional code. So specifically in your case, the test is a kind of is a good trade-off. You are like it's paying off. So to make the test.
Well, I mean, I guess the first thing I would say in my own experience is that it's just very difficult to get things correct without any testing. And especially if you have an application that you're anticipating will be complex enough that you're going to develop it over a long period of time, or maybe there will be many people involved. Perhaps the exact goals or the approach will change over time. If any of those are going to be true, then I think it's going to be important to have at least some amount of tests. The next thing I would say is that, there are a lot of different reasons why people do computation, and there are a lot of reasons why people build software. And some software is simple and some software is inherently more complicated. So let's say that you're developing a web application, which is just going to be like a straightforward display of data that's in a relational database. And in that case, you might have some code, but the code might be pretty close to almost being a declarative application. In contrast, consider like the development of a simulation algorithm for a mathematical simulation. In that case, there may be a complicated algorithm that you have to encode into software. And so in one case, I think you may be able to get away with a very small to close to no amount of testing, especially if you use something that's heavily typed or some framework which has some built-in validation and linting. In contrast, if you're going to develop scientific algorithms, I think in that case, it's probably going to be very difficult to escape the need for unit testing.
In integration, then
So,
end-to-end, and so on.
so I think like whether or not you need tests, it depends a lot on what you're trying to do. Like I think you always need tests, but I think the more complicated things that you build, the less graphical that they are, the less clear what the answer even should be, the more you're going to need tests.
Just one curiosity, how you end up doing like, because in general, when I see people work with system biology, biomathematics, bioinformatics, it's not very common to find someone like you showed a very strong movement between system biology, web application. I think that's the future. I mean, you should always think about to make web applications, so on and so on. How was this? I don't, it was like natural, or you in one stage of your life you decide like I need to start a big web application because that's important, or that was like natural, just like naturally you change from one side to the other. This like changing because you like a complete like soccer player. You do the model, you do the web application, people can interact with your web application. How that happen?
Well, I mean, just me personally, I mean, I've just personally been interested, and I've built software over many years, and me, being of a sort of newer generation, I've never really built much desktop software. So if I speak to older people in my field, or I look at simulation tools which were built earlier in history, they all emphasized desktop interfaces. Today, I think if you look at a lot of people that are starting new software projects, right? So I have the advantage of coming later in history. So if you're asking, partly why web and not, say, desktop, that's, I think, just a function of the fact that browsers have gotten better and there's more interest in general in web deployment. But
You said about React on the back end, because right now I'm reading a book about deep learning and the Node.js. Node.js is I like to program in Node.js. Do you have any experience with this heavy calculation? What are you using in the back end? Java, Node.js, what's in the back end?
So that's a good question. That's something that I wanted to mention. So you're asking about this intersection between web development and modeling, and so part of the answer to your question is just me personally. I have been involved in building web application, or what we've kind of come to know as web applications for a long time. And so it's just something that I am familiar with, and so for me, translating back and forth is not a problem. That said, of course, most scientists are not familiar with web development, software engineering in general, and certainly not web development. And so if you want to run a scientific team which is doing that, then I think someone in the team, someone has to have the vision that whatever gets built needs to be web accessible to make it accessible to the broader scientific community. But another dimension of your question is how do we actually do the computation? And so in our field, in systems biology, which is a little different than the machine learning, the community is smaller, and in our field, there's very little packages that are available in Node. So we do not really do any meaningful scientific computation in JavaScript or in TypeScript. the way that so for the Datonator project, for example, like all the data integration, all the data normalization, that's actually all done in Python. And then what we do is we deposit it, all the integrated normalized data into a Mongo database. Then the web application that you see, this just like pulls the data and displays it, but that's relatively simple. The complicated logic is all done as like a pre-processing step before you even get to the database. And then in the other project where we're using Angular, for example, where we're running simulations, in that case, all the logic is really encapsulated into the simulation logic is all encapsulated into Docker images. And inside the Docker images, we're generally running Python or C. In fact, there's no example yet where we're running JavaScript or Node more specifically inside of a Docker image to execute simulations. So, so the way that we basically make this work is that all the logic for simulations is hidden behind APIs, REST APIs, and those REST APIs are behind the scenes, either directly running Python, or for simulation, they're orchestrating the execution of Docker images, which are further hiding, encapsulating even more logic.
It seems that your solution was the same. I faced the same problem when I was doing my second postdoc. I faced the same problems. It seemed that your solution was the same. I didn't know. I thought to myself, how am I gonna do? So the solution I found was like yours. The Galaxy was run in a server, put the data available in MongoDB. MongoDB showed the data in the front end. But do you believe that maybe in the future, when someone start to make Node.js, because deep learning is one of the most difficult problems. So someone from Google wrote a book, a very nice book, that he said that JavaScript was made for deep learning. If deep learning is very powerful, JavaScript is powerful enough to run deep learning, I believe maybe it's just a matter of time until someone starts to build this package using JavaScript, because Node.js is JavaScript somehow. So maybe I think just a matter of time when you start to have a very powerful system biology like package using JavaScript. Do you think that it's a matter of technical problem? It's a matter of availability.
I mean, for the things that we work on, I think there's a few issues. So for this project, biosimulators and the related work that we call biosimulation. So in that case, one thing we're trying to do is we're trying to take all these simulation tools which have been developed, which can be used for different types of simulations, and standardize them to make them more reusable and composable. So in that case, what we're doing is we're taking advantage of simulation tools that have been created, some of which were initiated 20 plus years ago. And in that case, none of those are implemented using JavaScript. I mean, perhaps they could be today, but they were all developed and modelers made other choices. And so we're simply looking at that and observing that other people have chosen universally not to use JavaScript and Node. And so we just have to somehow embrace the decisions that they've made. And what we see is that to the extent that the community is aligning on a single language, that language is Python, not JavaScript. I mean, the other thing I would say is for that particular application, this is targeted at composing simulations together to construct what will inevitably be more computationally expensive simulations. And so I don't know that there'll be any reasonably near future in which it will be in which you'll really want to be able to run this in your own browser. So like you can forget about like this, separate these questions of whether you're going to use Python or C or Java or JavaScript to implement something versus where are you going to run it. So in my opinion, like if your goal is to run something in a browser, then of course like you need to figure out some way of translating most of the logic into JavaScript. But if you're going to run it on some remote server, then I think you don't need to necessarily implement it using JavaScript. You want it implemented in whatever way allows developers to develop it as quickly as possible. And I think in our field, at least, I would say it's substantially easier to try and build stuff in Python or maybe C++ because the underlying libraries that you need, there are many underlying libraries available to help you in Python. In contrast, in Node, there's just very little to work with.
Yep, I think that's maybe similar to a problem I faced with Java. Anyway, that's a very long question. I still on the same topic of innovative biomathematics. I would like to ask you a question because you are very close to the industry. So very close to the industry. You are in the science and the industry. Maybe specifically from the United States. Yesterday I was talking to a friend of mine, Carolina, and we were discussing about the difference between the industry and the scientific world. How they should, I don't know, it's in the United States is different because in Brazil there's a huge gap between the industry and academia. But I think they need each other. The one example is web application. Web application is a way to make available to the public something that was developed in the scientific world. Can you talk a little bit about how it works in the United States or other countries that had experience about the communication between science and the industry, the challenge that you face, because you seem very like easily moving between the industry and academia.
Well, I mean, I guess I can see that perspective, and where you might think of this as being close to industry, in the sense that there's software being developed, but at the same time, we don't have any collaborations with industry. We don't have any funding from industry. So in that sense, we don't really have much connection with industry, and I think there is a substantial gap between computational systems biology and models and industry. There's, I mean, I think there is a fair amount of use of flux balance analysis models by specific elements of industry. And there's certainly some use of modeling in pharmacology. But I think more generally, there's also still a lot of skepticism about the value of these systems models when in many cases they don't make particularly accurate predictions. So I think what a lot of industry is looking for is just still substantial improvement in the predictive capabilities of models. And until that point, I think the embrace of models will still continue to be relatively low, and they'll continue to be a relatively big gap.
So it seems that you are still very, we as a scientific community, we are still very far away to have a model that's going to be really used by someone. It seems that we are still very skeptical, as you said, about the validity of this model. It seems that those web application at the moment, they are more like academia, like stuff. Are still not being used by the industry, I guess, as you said.
I think where there is, and perhaps this is part of what you're reflecting on, is there's a lot of shared skills. So the kind of skills that are necessary to do this work are in high demand among industry, but people are using them in the industry for slightly different purposes. And so that is kind of what I think creates this sort of feeling like there's a connection or close connection with industry, even though industry is not actually using these kind of models. But so that does have interesting consequences, though. So one consequence of that is that this kind of work might actually be quite good preparation for work in industry. So, for example, like your typical computational biologist has probably never heard of testing, but in order to build a large model or large software application to support modeling, you probably need to learn about testing and continuous integration and code management and so on. And those things, those skills, I think, are much more heavily prioritized by industry than academic science. The other thing that we see in the United States is, as a result of that, and that there's also a big salary gap between academic science and industry, which is probably true of most of the world. It's not that.
But the gap is in favor of the industry or in favor of the academia?
Academia is industry pay is much more favorable, and so that can make it difficult to find enough people in academia with the skills that are needed to do this kind of work.
Okay. I would like to maybe to close the discussion with two questions from social network. I have one is from LinkedIn. I student from a PhD student from mathematical biology. He asked me the following question, but you already answered that, but just to summarize. So he said, like, I'm in the first year of my mathematical biology study. I'm a little bit lost. Could you please give me some advice by what should I start and which are the skills that I must learn? He's talking about the skills that you need to start to be a good mathematical biologist. You already said something about that. Can you maybe summarize the best skills that someone must have if they wanted to work with mathematical biology?
Well, I mean, the first thing I would respond and point out is that mathematical biology is a very broad field. And so I don't want to overgeneralize about what skills are needed. The skills that are needed for different areas, I think, can be quite different. So, for example, if you wanted to work in genome informatics, in that case, there's a lot of established methodology, there's a lot of established software tools, and so a lot of people focus quite a lot of time on learning how to use very specific established methods and tools, and there's a lot of emphasis there on assembling those established tools into workflows. In contrast, in computational modeling of dynamical systems, the kind that I described, I would consider this a comparatively less mature field. And in that case, I think there aren't so many software tools or established methods that someone should really prioritize trying to learn. And in that case, I think instead, it's really important to try to develop more foundational skills in computation. And it's I don't mean to suggest that the computation is necessarily the most important skill to learn. In my experience, it's a frequent set of skills that many early graduate students, many postdocs just have not developed, because I think a lot of schools, a lot of programs in and around computational biology, they focus a lot on development of physical concepts, mathematical concepts, but not so much on and basic introduction to computer programming, but they don't focus a lot on how to build complex computational systems. And in contrast, the practical work of doing computational biology, especially in an immature area, involves a lot of data management, software management, And so what I've seen is a lot of people, I think, start to get stuck, or they start to their work becomes extremely inefficient because they haven't been taught how to more systematically or rigorously deal with complex computational systems. So that's why I emphasize, particularly for more immature fields, that I think it's important to really build a strong foundation in how to write high quality code quickly. And I think for a lot of beginners, unit testing, and also if you're working in highly complex mathematical codes, I think learning how to do unit testing and continuous integration is probably some of the most essential practical skills that you could learn, and relatively simple.
Okay, but would you say, because you said something very nice, that there is a kind of problem in the education system, that they focus too much on the mathematics. MIT is very, I saw that you had a very, I think that you did your PhD, if I right, in the MIT. And the MIT is very famous because they're always trying to think. In your case, MIT was helpful in that sense to you, or you believe that they also needed to change a little bit how to handle this kind of situation?
Um, so I was an undergraduate student at MIT, and I was a graduate student at Stanford, and I took a lot of courses at both institutions. And I can tell you in my experience, first of all, it was several years ago, so it's possible that the curriculum has changed, though I don't think it has. Undergraduate students, even at those institutions, and graduate students, generally will not be exposed to things like unit testing, continuous integration, even most students working in computational science. As an undergraduate student, so the only students who are really exposed to, there's only really like one course as an undergraduate student at MIT that really exposes people to things like unit testing, and it's a software engineering lab, and it's really only software engineering students that take it. Computer science students, more specifically, that take it. Generally, I mean, maybe some students that are earning multiple majors will take it, but like students like myself who are interested in like computational physics, computational biology, bioinformatics, generally don't take that course. They would usually take courses in like computational systems biology, algorithms, data structures, but they don't usually take these lab classes because the lab classes are a huge amount of work. Usually the folk, like, in my experience, was that at MIT there are certainly people who will take like 6-7 classes or more a semester, but it's very unusual that somebody's going to take more than one lab class, because the lab classes just are a huge time sink.
Yeah. Ernst was smart. He didn't take class. He used to take the note of somebody else.
So what I've seen, and also if I talk to I have a number of friends from college who are software engineers. I mean, even the ones that took the software engineering class, I mean, they'll largely tell you that they didn't learn how to test code through that class anyway. They really learned on the job after they graduated, or maybe during internships during college.
My case as well.
And so that's really how I learned about all of this. There was a brief period of time. One of the most informative periods for me was that there was a brief period of time where, as a graduate student, I worked with a guy who had previously worked at Google, and then, but more immediately, he had worked at a different fintech startup company. And so I really learned from him really on the job. I didn't learn all of this through coursework. And even retrospectively, knowing what the concepts are, now I can confidently say that they're basically, aside from this one software engineering class, there are no classes that I could have taken which would have introduced this. And at Mount Sinai, for example, we actually do have a course which introduces this, but very few students take it.
Okay.
And I think that course is relatively unique. I mean, we do offer it here, but not many schools offer something similar to that.
Yeah, that's interesting.
So I think it's a topic which is quite important to the practice of science, of complex computational systems, which arises both in software engineering and in computational science. And it's something which is typically learned on the job. And one thing to keep in mind is most of the faculty who are teaching courses, they don't know about this topic, so of course they're not teaching it. The people who know about it don't work in academia. The people who know about these things, they work in industry.
So you are saying that the people say that who knows is doing, who does not know is teaching. Have you ever that?
Well, I think it's more that academia and industry, they focus on different problems. And so the problems that people are working on in industry, they have driven the development of a certain skill set. And academia, by virtue of working on different things, doesn't necessarily foster the same skills to the same degree, even though on the surface, there's a lot of similarity between academic research and particular aspects of engineering.
Okay, Jonathan. I just want to ask you a last question. This one is from the Twitter. One of the that participate on Twitter, he asked, what is needed for one to be called a biomathematician? If you want to call someone a biomathematician, what is needed to be called? I don't know the answer. Do you? Because
I mean, there's no certification. There's no formal definition of what a biomathematician is. I mean, I guess one thing I could say is I think the lines around biomathematics, the lines around computational biology, bioinformatics, these are very blurry lines. Systems biology also, these are kind of gray areas. I think anybody who combines some math and computation with biology could potentially consider themselves a computational biologist or a mathematical biologist.
But do you think that's something bad or that's something good? Because do you think that we should define better the boundaries, or it's better to leave it the way it is? Let people do whatever they want and call themselves biomathematician, or system biologist or whatever.
I mean, for the most part, I think I don't feel like there's a strong need to have labels. I think that different scientific problems require different skills, and I feel like the key thing is that whatever scientific problem that you're interested in, that you should figure out what skills are essential for that problem and try to go develop those skills. And maybe they'll be computational, maybe they'll be mathematical, maybe they'll be neither of the above. I think where the lack of definitions is a little bit problematic is so in systems biology and then on other aspects of biomathematics, computational biology, there are these aspirations to have really big models of biological systems, and to the degree that requires collaboration, and that in turn is probably going to require concentrated funding on a few specific problems. I think that this community side of coalescing people around individual problems or individual projects, that I think is challenging when there's no clear definition of what the field is. And so without clear definitions, the field is very diffusely scattered across a large number of questions. And so that means many people are simultaneously investigating different things. And so from a certain perspective, that's good. But it also does mean that it's, I think, difficult to assemble a large team of people around any one really big problem.
Yeah.
And
I
so
agree.
that is to some extent at perhaps the detriment of large-scale science.
Okay. So, John, thanks a lot. That was amazing talk. I could go on for hours, but I don't want to take more of your time. And you are also invited to come back in the future to bring a new topic. I think that's one of the best talk, one of the best live, because I already knew that because I saw your talk previously in Como Lake, and I know how nice it is. So I would like to thank everyone for participating. You have all the information of Jonathan in the description of the video. If you want to leave an email, he can leave me. I'll put in the description of the video. So, Jonathan, thanks a lot, and we'll keep in touch.
Yeah, thanks so much for organizing this.